Hello and welcome to the penultimate Bio43 pre-lab video. This week, we'll go over the steps required to clone your gene of interest and use some computational tools that are important for planning out the cloning process. Earlier, you selected a gene you want to study. The goal for this unit is to produce a large quantity of the protein encoded by that gene so that you can analyze it and learn about its function. We'll use E. coli bacteria to produce the protein for us, so we need to put your gene of interest into E. coli cells. The first step of that process is making a lot of copies of your gene. PCR, or polymerase chain reaction, is an excellent tool for this. PCR mimics the steps of normal DNA replication, but it replaces the need for most of the replication enzymes using temperature fluctuations and small pieces of single-stranded DNA called primers. This is a technique better illustrated with animation, so I've put a video about PCR on the lessons page that has some nice art design. The polymerase and nucleotides needed for PCR are commercially available, but the template DNA and primers you need will be unique to your gene of interest. For your template, you need a piece of DNA with your gene on it. Genomic DNA is not a good source since it has introns that E. coli won't know how to remove. A better option is to start with the mRNA, which already has the introns spliced out. That means we need a source of cells that express your gene of interest. For non-human genes, this usually isn't too hard to find. But for human genes, there are ethical considerations. It may be possible to find existing cell cultures that express your gene, but you might need primary human tissue. Many hospitals have research departments and use patient tissue for their investigations. Take the example gene from my last video, MEFV. Based on its expression pattern, it looks like the appendix would be a great source of mRNA. Conveniently, many appendices are removed and discarded every day. However, researchers, even those working at hospitals, can't just take whatever medical waste they want. There is a dark history of scientists taking patient tissue without their knowledge or consent, and worse. As the next generation of scientists and medical professionals, it's important to be aware of this and work to do better. Today, any research with human subjects requires the approval of an institutional review board, and most uses of tissue samples require the donor to be fully informed of the intended use and to consent to that use. There is ongoing discussion about how to properly compensate donors and maintain their privacy. So let's say we've obtained some appendix tissue through the proper ethical channels and purified the mRNA from it. Unfortunately, PCR doesn't work with mRNA, so we need to convert it back to DNA. There's an enzyme called reverse transcriptase which does just that. The product of this reaction is called complementary DNA, or cDNA. This cDNA will be our template for PCR. What you recorded as your mRNA sequence last time is really cDNA, which is why it has T's instead of U's. Let's put that sequence into a program that will help us plan the rest of our cloning. Benchling is a convenient online tool that is free for academic use. Register for a free account before lab this week. I'll walk through the steps you'll do in class. First, create a new project. I'll call mine MEFV Cloning Demo. Give it a brief description. You can add your group mates, TA, and instructor as collaborators. Now create a new DNA sequence. This will be the MEFV cDNA, and this is linear. Add this to the project we just created. Click Create. Now we'll paste the FASTA sequence here. You can see that Benchling has added a lot of information. The other DNA strand, numbering, and these are restriction sites. Places where enzymes called restriction endonucleases, or restriction enzymes, can cut the DNA. More on them later. So this is our template, but the mRNA, and therefore the cDNA copy, has more than just a protein coding sequence. There are regions upstream and downstream of the translated portion that we don't want to amplify when we do PCR. We can go back to NCBI to determine which part of the sequence codes for the protein. Previously, we ignored this page and went straight to the FASTA sequence. But there is more useful information here. Basic summary info, references, but the key information we want here is features. This tells us that the coding sequence, or CDS, covers bases 47 through 2392. To keep track of that, we can annotate it in benchling. Highlight any sequence, create, annotation, give it a name, input that range we just found for position, forward orientation, choose whatever color you'd like, and save. Click on the newly created annotation and choose Create, Translation, Forward to display the translated amino acid sequence. Now we've marked the region we want to amplify using PCR. But before we design our primers, we need to know a little more about the restriction sites that are present. For our project specifically, we need to know about the sites for NDE1, ZO1, and BAMH1. Benchling displays a lot more than that, but we can narrow it down. Click on the scissors on the right. Change the settings so that no cut sites are visible except selected ones. Then search for and select NDE1, ZO1, and BAMH1. Ideally, at least one of these sites will not be present in your gene of interest. We can see there are NDE1 and BAMH1 sites in MEFV, but no ZO1 site. There is a ZO1 site on the cDNA, but it's outside of the coding region. We'll keep this in mind going forward. Now we're ready to design primers. Start by highlighting about 20 bases at the start of the coding sequence. We actually don't need to include the start codon, so we'll begin after the initial ATG. A good primer is between 18 and 30 bases long, has between 40 and 60% Gs and Cs, has a melting temperature between 60 and 65 Celsius, and ends with a G or C. The length, GC content, and melting temperature can all be seen here at the bottom. Currently, the melting temp is a little low, so let's extend the selection a bit in the forward direction. Here we have a sequence 29 bases long, 55% GC content, and a melting temperature of 63 degrees. Looks good. So we go to Create, Primer. This will be our forward primer. This sequence would be sufficient to amplify the gene with PCR, but we're going to add a little more. Any DNA sequence put on the 5' end of the primer will be added to the end of the amplified gene. I want to add a ZO1 cut site. I can get the sequence of the cut site by searching for it here, and add it to the start of the primer. For this part, you can add any cut site that doesn't appear in your cloning sequence. But if you're using BAMH1, add an extra A between the cut site and your gene. This step is not necessary for NDE1 or ZO1. Before the cut site, add 4 arbitrary bases, GCAT. We'll discuss the reasons for this later. Name the primer, keep it in this project, and save. Making the reverse primer is similar. Select about 20 bases at the end of the target region. We will include the stop codon here. Melting temperature is a little low right now, so we can extend. This time to the left. We want the two primers to have melting temperatures as close as possible. Just below 64 degrees looks good. GC content is a little higher than ideal, but not terrible. Create, primer, reverse this time. Add the ZO1 site at the 5' end, and the 4 arbitrary bases before that. Name, and save. Now click over to Pairs. Back in the sequence, select the forward primer, then scroll to the reverse primer, hold Shift, and click on it. Then choose Link Primers to let the program know that these will be used together in PCR. Choose Create PCR Product to make a new DNA file. This is the sequence of the PCR product you'll make with these primers. Go to the project icon on the left to rename it. There are companies that will make us custom primers with these sequences. Mix them with the template DNA, polymerase, and nucleotides, and we can complete our PCR. So now we've got billions of copies of our gene of interest. But if we were to put these pieces of DNA in E. coli, the bacteria wouldn't know what to do with them. So first, we're going to put the gene in a plasmid. A plasmid, also called a construct or vector, is a small circular piece of DNA that is separate from the genomic DNA, but is still replicated and passed on during mitosis. Let's take a look at the plasmid we'll be using, PET16B. Create a new DNA sequence. This one is circular. Paste in the sequence. Change to plasmid to see it in the circular form. I'll annotate this sequence so you can see the important features of the plasmid. The origin of replication, or ORI, is where replication of the plasmid begins. Two protein coding genes are already present on the plasmid. The first, BLA, grants the host cell resistance to the antibiotic ampicillin. The second, LAC-I, will be important for regulating the expression of our gene of interest. The multiple cloning site is where we'll be inserting our gene into the plasmid. You can see it contains the same 3 cut sites we discussed earlier. Near the cloning site, there are several important features. Upstream, there's a T7 promoter. An RNA polymerase will bind here and transcribe what follows until it reaches the T7 terminator. So if our gene is inserted in between those sites, it will be transcribed. The LAC operator works with the LAC-I gene product to ensure expression only occurs when we're ready for it. More on this next week. Then there's a start codon where translation will begin, which is why we didn't need to include the start codon in the PCR product. This is followed by a hist tag, a sequence which translates to several histidine amino acids in a row, and a factor XA site, a sequence that translates to a protease cut site. More on both of these next week as well. So we want to insert our gene at the multiple cloning site. This is where restriction endonucleases enter the picture. These are enzymes that cut DNA at specific sites, typically 4 to 8 bases in length and palindromic. That is, they read the same 5' to 3' on the top and bottom strands. CTC-GAG is a site where the enzyme ZO1 cuts. It cleaves the DNA backbone between the first C and T on both strands. So after cutting, or digesting, the plasmid, there will be overhangs of 4 unpaired bases. We added the same cut sites to the ends of our PCR product, so we can cut that with ZO1 as well. The extra bases we added to the ends are important because some restriction enzymes don't function well too close to the ends of linear DNA. After we digest our PCR product, it now has overhangs that match those of the cut plasmid. If we mix the digested plasmid and PCR product together, the overhangs can pair with each other. And we can use an enzyme called DNA ligase to seal the backbone. With that, we've successfully inserted the gene into the plasmid. To see what this plasmid will look like, first make a new copy of the PET16B file. We'll call this PET16B MEFV since it's the joining of these sequences. Go back to the MEFV PCR product, select everything except the 4 extra bases we added to each end, copy, move back to the PET16B MEFV file, highlight the ZO1 cut site, and paste. If everything went right, the reading frame for your inserted gene should be the same as the pre-existing one in the plasmid. The ZO1 and NDE1 sites are already in frame. The 6 bases correspond exactly to 2 codons. The BAMH1 site isn't aligned like that. This is why I said your primer has to have an extra base between the cut site and the start of your gene when using this enzyme. If your sequence doesn't align nicely like this, you have accidentally added or forgot an extra base at some point. Review your work carefully. So that's the idea. Prepare your template DNA, design primers to amplify your gene of interest with the proper restriction sites added, perform the PCR, digest the PCR product and your plasmid, combine the pieces, and ligate. If you join a molecular biology lab, you'll find there's a little more nuance, and a lot of potential troubleshooting if things don't go perfectly at first. But this is a basic framework for cloning your gene of interest into a plasmid. And this is really how we plan out the process in the lab. Next, we'll put this plasmid in E. coli, express the protein, purify it, and start using it in experiments. See you next time.